long-chain circular DNA, such as plasmids, bacterial and viral genomes. How to produce such a large circular DNA? Today, E. coli cloning is widely used for amplification of large DNA. It is an established and viewed as an effective method. But in fact, it is a cumbersome and time-consuming method requiring skilled techniques. It is difficult to cloning cell-toxic sequences. Also, there are concerns about biosafety. Our new product, OriCiro Cell-Free Cloning System, is now on the market. It contains assembly kit and amplification kit. Multiple fragments can be assembled efficiently in the assembly part by adding the assembly product into the following the amplification reaction. You can get a large amount of circular DNA by a mechanism of chromosome replication. The beginning of experiment, you should design and synthesize gene fragments. It can be a PCR fragment or a restriction enzyme cleavage fragment. For the amplification reaction, you should design including the OriC fragment that will be the origin of replication. Step 1. OriCiro Assembly Technology. Put your DNA fragment into the assembly solution. The OriC fragment, which is the origin of DNA replication, is also added to the assembly solution. Incubate at 42 degrees for 30 minutes. In the OriCiro assembly kit, 20 to 60 BP overlap sequences that are homologous to the fragment, to be linked are designed at both ends of each gene fragment. One side of the double strand is digested, exposing the single strand containing the overlapping sequence. This kit efficiently annealing with enzymes. You can assemble up to 50 DNA fragments into a large circular DNA with a single step, enzymatic reaction. And for the subsequent amplification reaction, gene design should be circular DNA. We know that the reaction solution after ligation contains fragments other than the target circular DNA. The step of selectively preparing only circular DNA is the next amplification reaction. Step 2. OriCiro Amplification Technology. Ligation reaction solution from Step 1, directly added to OriCiro Amplification Kit, which contains 26 proteins essential for E. coli genome propagation process. Reacted at 30 degrees Celsius for 3 hours or more. Only circular DNA is selectively amplified, and linear DNA are not amplified. First, the enzymes starting from OriC, for DNA replication. Replication proceeds in both directions from OriC. When the replication is completed, the next replication will continue using this as a template. The circular DNA that has been replicated to 4 circular DNAs. Continuously, the circular DNA is amplified from 4 to 8, and 8 to 16, in an exponential manner. Finally, only the desired circular DNA is obtained in high purity. OriCiro Amplification Kit provides up to 1 microgram of supercoiled DNA can be obtained by a reaction of 10 microliter. 104 times more accurate than PCR Taq polymerase. Up to 50 kilobits of circular DNA can be amplified. Applicable to all sequences which cloning is difficult using E. coli. For example GC-rich sequence. And can be used directly for transformation of E. coli. OriCiro Cell-Free Cloning System is now on the market.